Excellent. Welcome, everybody, to one more session. It's a real pleasure to have with us Catie Chang from Vanderbilt. Catie did her bachelor's at MIT, master's and PhD from Stanford, a postdoc from NIH, and then she has been faculty at Vanderbilt since 2018, right? And yeah, so as usual, we're looking forward to the talk. And as usual, if there are questions, you can put them in the chat. I will be monitoring the chat. So all yours, Catie, go for it.
Sounds great. Yeah, thank you so much for this opportunity to talk with you. I'm going to share today some of our research directions on studying dynamic brain states using fMRI and other complementary biological signals. And so, just a little bit of background, fMRI is capturing dynamic fluctuations in blood oxygenation over time, which is an indirect measure of neural activity. And since we are measuring a vascular physiological response, this is limiting our temporal resolution compared to techniques such as EEG, which more directly capture the brain's electrical activity. But a great advantage of fMRI is our ability to achieve millimeter scale spatial resolution throughout the entire brain, including within deep subcortical structures. And so this provides very rich data. So we're able to map these large-scale patterns across the brain that capture the functional organization of different regions, as well as how different brain areas are interacting with one another to form either local or distributed networks. And we can also, of course, track how this activity is behaving in time, and the temporal dynamics of fMRI have been gaining more and more attention. And so fMRI is in fact a spatiotemporal data set, and if you just watch the patterns in the data, you can observe very interesting dynamic structure. And even if the person that you're scanning is just quietly closing their eyes and resting rather than engaging in some specific behavior or task, you can still see a lot of this structure. And so that's really pointing to how much organization there is even just in the endogenous spontaneous activity of the brain. And so this has prompted the field to ask questions like, what is the functional relevance of these temporal variations in activity across the brain? And relatedly, can we capture features from these dynamics that can provide useful clinical biomarkers? So maybe something about regional dynamics or the coupling between different regions over time. And so there's been a growing amount of work in the computational methods for extracting relevant dynamic features from this data, and also in trying to map these kinds of dynamic features onto individual differences and disease. And by the way, if anyone has questions during the talk, please feel free to interrupt or ask questions on the chat. And so, yeah, going forward in that direction, a number of studies have been pointing to this idea that for certain cognitive or clinical traits, the fine-scale temporal behavior in fMRI signals may provide potentially more sensitive information compared to measures that capture longer time averages of either activity or network connectivity. So I'll just highlight one of the studies here where they found that faster timescales of fMRI dynamics seem to be better indicators of cognitive and attentional assessments compared to if they were looking at longer, what they call static time averages, although not always. So there were some measures where the longer time averages seemed to capture more variance in the behavioral or a trait measure, including some of these lifestyle or self-report qualities. So as an example of a clinical application, this was one study that I worked on in my graduate studies in collaboration with Ian Gottlieb's lab and Paul Hamilton at Stanford. And they were studying major depressive disorder. And what we did was we took these major brain networks, a default mode network and dorsal attention network, and we extracted time courses from these areas. And by the way, one of these networks is thought to be more involved in internal self-referential processing, that's default mode network. And the dorsal attention system is thought to be more involved in an externally driven processing or cognitive control. And so we extracted the signals and kind of looked at their relative dynamics and just captured a simple measure of how often the subjects seemed to spend time in a state that was more dominated by one network than the other, and found that this actually happened to correlate well with measures of rumination that the subjects would report after the scan. And so specifically, the more time a subject seemed to spend in a state that was dominated by the more externally oriented network, the more the person tended to engage in more of a reflective or constructive form of rumination. Okay, so the studies that I just discussed, they tried to capture some measure of fMRI dynamics, but use those to predict more stable trait-like measures. So typically those trait measures are collected separately from an fMRI scan, for instance, by using questionnaires or neuropsychological tests. But another question we could ask is, to what extent the ongoing dynamics of fMRI signals are reflecting a more instantaneous functional state of an individual? So in other words, can we look in fMRI data and track on more of a real-time basis the kinds of states that people might be experienced during the fMRI scan itself? And indeed, there have been studies pointing to the fact that fMRI can give us interesting measures like whether or not we'll perceive an incoming stimulus that's just barely perceptible. Maybe sometimes you perceive it, sometimes you don't, and the patterns in your fMRI activity may be able to tell how you'll respond. Similarly, changes in cognitive states or activities like mind wandering. And so we may be able to probe these important internal states by investigating these time-resolved fMRI patterns. And this might in turn give us more tools for understanding how these kinds of states may be altered in disease. So along this theme, I'm planning to share a few studies in two related directions. So the first is on fMRI signatures of dynamic states, focusing on vigilance or changes in wakefulness. And the second explores close connections between the brain and the body. And for this, for both studies, we're going to combine fMRI together with measures of brain electrical activity and physiological measures. So as I mentioned, we've been particularly interested in vigilance, and I'm going to use this term to describe natural drifts in alert wakefulness to drowsiness and light sleep. So this phenomenon is also referred to as arousal. And one reason this became really interesting to me is that in practice, when you're doing your fMRI scans, people often get sleepy and they fall asleep. And we may or may not know that. I always fall asleep when I'm a subject, so I'm the worst subject to ask for volunteering for your study. But the other, one of the practical implications of this is in our data analysis. So when people are transitioning between these major arousal states, this is reflected in changes in the characteristics of our brain activity and our physiology, which becomes which translates to what we see in our fMRI signals. So, oops, did someone say something? Oh, sorry. Yeah, feel free to interrupt me if there's any questions. I can't see the chat right now. Yeah, so in one sense, we might say, these vigilant state changes are kind of confounds in our studies, right? It's kind of annoying if we can't tell when subject is asleep or not, and maybe their behavior is different during certain periods and their brain activity, and we have to model that. But on the other hand, I really don't think we should think about this purely as something confounding our studies, but also a really interesting aspect of brain function that we can study. So vigilance regulation is altered in many neurological and neuropsychiatric disorders. And of course, there's really interesting interactions between your vigilant state and how you are perceiving information in the world and how you behave. And so if we can see fMRI markers of vigilance levels in our data, then this also gives us powerful tools for trying to study this in itself. So, some of the questions that we've been asking in this research are firstly, if you have a change in vigilance, how is that reflected in whole brain fMRI patterns? Can we detect changes in vigilance states directly from fMRI data? And do we see any connection between the spatial or temporal dynamics of vigilance signals in fMRI and clinical measures? So this first study I'll show was the idea of a student, Catie Chang, who was a PhD student in our lab. And he had the idea to test the extent to which vigilance levels are emerging in low-dimensional representations of fMRI data. And so specifically what we did was we took the whole brain fMRI activity at each individual time point. So in fMRI, you're getting these 3D volumes over time. For each time point, we can divide that whole brain fMRI pattern into different regions of interest to do some kind of initial dimensionality reduction. And then we took these vectors and projected them further into lower dimensional space, and we experimented with different techniques such as PCA and UMAP. And then what we were interested in is, well, each one of these time points then gets, represented as a dot in this lower dimensional space. And we wondered if this, the organization aligns in some sense with the vigilance level of a person. So during this kind of scan, subjects were actually performing a reaction time task. And so for every time point, we had a behavioral measure, corresponding to the closest trial in which they made a response. So they had to respond as quickly as they could, and then we could color these points by their likely alert or drowsy states based on how quickly they responded to the task, and ended up seeing some interesting structure in that. And that there was somewhat of a gradient across the space in which more drowsy or more alert time points seemed to cluster. At the same time, we also recorded EEG. So EEG has been used for quite some time in studies of sleep, for example. There are clear measures of vigilant state in the oscillations that you can see in the electrical activity on the scalp. And so we also tried to confirm these patterns by coloring now each point by their EEG index of alertness or drowsiness, which is based on spectral power in alpha and theta frequency bands. So then, in addition to kind of looking at this, distribution in a low-dimensional space, we just clustered the time points. So we just said, let's kind of discretize this. We didn't, it's possible that clustering is not the best thing to do, but we thought, let's just see what happens if we try to form, say, two clusters. And what we found was that, if this is a scan over time, in each one of these, this is kind of the assignment to clusters. We could see in this particular scan the assignments to the two different clusters, and when we superimposed the EEG spectral measure of vigilance, we could see quite a good alignment with that, which we thought further supported the fact that fMRI patterns in a low-dimensional space may be reflecting information that's relevant to one's vigilance levels. So we don't really get any information about brain anatomy from this kind of representation, right? So we've kind of collapsed information down, and we're not really looking at, which brain areas are doing what across these different states. And so what we did to take another look at that was, or a further look at that, was basically just to average the time frames that, or actually, sorry, further, cluster the time frames and look at their spatial patterns. So the first thing we did was allow there to be additional clusters, and we were actually curious if these nested within the larger alert and drowsy states. And then for each one of these kind of substates, we would look at back in the original space to see what the fMRI patterns appear to resemble. And so we saw, during higher arousal states, there tended to be more clear network patterns. If you're used to looking at fMRI data, you might recognize networks like the default mode network or dorsal attention network that seem to be anti-correlated with each other, which is the different colors. And in a drowsiness, it seemed that there were tended to be more activity in primary sensory cortices, and there was not quite as much anti-correlation between the cortical systems. And so this tended to be consistent with prior observations in the literature, using some indicator of states, brain states as well. So we then were interested in, looking further at whether these more fine-grained stub states actually do nest themselves into the coarser-grained alert or drowsy states. And so to do that, we started to look at the temporal transitions between these substates. And so in this figure, we allowed there to be 15 different clusters, and this is representing the state transition matrix. So the IJ entry is estimated by the fraction of times that being in state I at one time point moves to state J at the next time point. And what emerged from essentially clustering this transition matrix was that there were two main groups of these substates, which indeed seem to correspond fairly well with the two more coarse alert and drowsy states, which are colored the same way as they were in the previous slide. And so these, we're all kind of transitioning to one another in the more drowsy state, whereas these tended to be within the more alert state. But interestingly, there were also some kind of bridge states that tended to be outliers in their own community. So some of them actually showed a tendency to transition more to the other group. So that was quite interesting. So overall, I think one thing this work highlights is that vigilant states do come with differences in the activity structure in fMRI data, and that we can see that in the low dimensional representations. It also shows that we can detect vigilance information in fMRI in a pretty time resolved fashion, and even in individual subject scans, possibly even in single time points. And it also suggests that there may be value to parsing fMRI scans by these brain states and maybe looking specifically at the repertoire of patterns that are associated with these different states. So in addition to looking at these more discrete types of vigilant states, we were, and quantized, I would say, we were also interested in looking at whether fMRI data can actually track more continuous levels in someone's alertness or vigilance over the scan. And so the question that we posed here was, given an fMRI data set as input, can we essentially output a one-dimensional time course that is estimating, how alert, how drowsy that person is at a given moment of time during the scan? And we thought this might be useful for a couple of reasons. One is that because it's cumbersome to acquire EEG, maybe eye tracking, and other gold standard measures of someone's vigilance during fMRI, then if we can read out this information from the data, then that might be useful for understanding how we should model it. And so the approach that we took here is actually more of a supervised learning approach in contrast with the previous study, which was completely unsupervised. And what we do is we have recorded simultaneously EEG along with fMRI in a subset of our scans. And we use that to learn a whole brain pattern of fMRI data that tracks increases and decreases in an EEG measure of vigilance. And so then the idea is that if we've learned this kind of a priori pattern, given a new data set that may or may not have EEG recorded, we would then project that pattern onto every successive fMRI volume of the new scan, which produces a scalar value. And over time, that traces out what we took to be an estimated fMRI alertness index. And here we did overlay that with an EEG alertness index because we wanted to validate it first. So we used a validation testing set that also had EEG. One second. And so, yeah, one of the hopes for this kind of approach is that there's also a lot of data recorded and shared already that don't have these EEG or pupil measures. So it would also maybe give us another way to query that kind of data. Incidentally, our first study in this direction was in non-human primates, just because I happened to be working on a project where we were scanning awake macaque monkeys, and we had also recorded their eye behavior, which can be another indicator of their vigilant state. And in this data set, and in some of the monkeys, it was actually very clear that they would get very bored and sleepy and they would start dozing off. And I would watch these videos of their eyes and it would make me really sleepy. So I could, at the same time see quite substantial changes happening in fMRI data as they transition between these putative like alert or drowsy states or sleep. And so, yeah, we also tried this kind of approach in this data. And so here was like the monkey's tendency to spontaneously have their eyes open or closed during the scan. And if we use our approach, we estimated based on their fMRI data the time course of when we of their potential vigilant state. We also validated this with measures of intracranial electrophysiology. So the monkeys also had electrodes in specific brain areas, and we could also find that this estimated fMRI index could track the electrophysiological measure of vigilance as well. So going back to humans, the next question we asked was whether this could potentially be useful in explaining variability in behavioral performance. So in fMRI scans, we might be presenting some stimulus to a person, and even if you show the same kinds of stimulus over and over, there's still variability in human behavior. So sometimes we might respond, if we're asked to respond as quickly as possible, sometimes we might push the button very quickly, sometimes for various reasons it might be slow, sometimes we'll miss it. And we use this kind of very simple task as a test bed for this. So in this case, the person was hearing an auditory tone, and the tone was presented at like kind of long stimulus intervals. So you might hear a tone and then only 30 to 40 seconds later would you hear another one. And so with this kind of design, we thought that much of the variability in someone's reaction time might relate to, how sleepy they are. And so we then took a small window of time before each stimulus, so like 5 seconds before a trial happens. And we looked at our estimated fMRI alertness index and said, can we predict if a few seconds later, if they're going to actually make a response, or if they're going to miss it, or if they're going to make a slow or fast response. So this is a little bit more about our data, if in case you're interested. So this is multi-echo fMRI acquisitions, and again, we did very long. Actually, these are long scans by fMRI standards because we were hoping to see if we would get some changes in someone's alertness across time. So this project was led by Sarah Goodale, also a recent PhD graduate in our lab. And here we are showing the trials where a person responded versus when they missed it altogether. This is indicating that the estimated levels of alertness from this fMRI measure tended to be higher in each person when they were, in most of the people anyway, when they made a response versus when they missed the trial. And then if we kind of zoom in to look at what's going on around each individual tone. So if time 0 is when the auditory tone was presented, we can then average what's happening in our fMRI alertness index around the time of the trial. separating what happens when for the trials where a response was made compared to when it was missed. And so you can again see this kind of divergence happening even before the onset of the trial. We also noticed this kind of interesting bump afterwards, and one of our speculations is that sometimes the tone would actually wake a person up. And so if they were sleeping or starting to get sleepy, they might have a bit of an increase in their alertness afterwards as well. And because we had recorded EEG data at the same time, we could also look at spectral EEG spectral signatures of alertness, which, as I mentioned before, are better understood than fMRI at this point in time. And so this also validated that during the trials where the subjects missed the response, they had more power in their low frequency of their EEG signals, suggesting that they were indeed more drowsy or even asleep. So we then looked, at the trials where a subject did respond and separated those into fast versus slow responses, and again saw this trend toward being able to predict. This is even before the stimulus comes on. Is where we're making the prediction. Whether or not the person will respond quickly or slowly, and also just looking at an aggregate across the trials across all subjects as well. And these are the sort of binned, fast, slow, and then missed trials. So this study as well also indicates that fMRI may be sensitive enough to track changes in vigilance over time. So not only can we kind of see some change in an fMRI pattern, but it might be actually reliable enough to allow us to make a generalizable tool to do this across different people to estimate their vigilance levels. Right. Yes, and we also had, demonstrated that this may correlate with measures of behavior and with the electrophysiology. And again, some implications for studying brain function, and in particular studying individuals, which is one trend that the field is moving toward. If we're able to understand vigilant states in the brain and we can model their effects, then this should allow us to obtain more sensitive and interpretable inferences in terms of whatever we're studying with fMRI, and hopefully with less data, since we'll be able to account for the source of variability. And then we don't want to see this just as some kind of confound, but we've actually been looking into whether or not these fMRI vigilance signatures could provide new individual markers of interest in cognition and disease. And so I'll show you a couple of studies along those lines as well. And so, Kate.
Can I ask a couple of questions?
Oh,
It's a
yes,
good
yes.
time to interrupt. So if I understand correctly, the fMRI-based alertness index was derived by training it on the EEG-based alertness index. Is that right? You had some training paradigm to find the best.
Yes, so it was trained using concurrent EEG and fMRI data.
So
And
you
so
tried to capture the EEG index from the fMRI, right?
that's
Would
correct.
would this work even if the subject had their eyes closed throughout?
Yes, and in fact, I forgot to mention that the subject does have their eyes closed throughout the entire scan for the human study that I mentioned. So for the monkey study, we couldn't instruct them to close their eyes, and they weren't
Okay.
trained to do that. So they were just kind of keeping them or doing what they wished. But for the humans, we have their eyes are closed in all of these experiments. And the EEG is serving as our indicator of their brain state, since we can't really use their eyes. Yeah, thank you for asking. Oh, I see a question also. How long does it take to determine an fMRI vigilance measure? Is an fMRI noisy? Yes, fMRI is noisy. Actually, the signal to noise ratio is not great, but if you try to pre-process the data as best as possible and remove other types of artifacts, head motion, etc., then it can become much cleaner. We were trying to determine these measures on an individual time frame basis. So that's kind of tracing out this waveform. And there's probably, noise in this waveform. Let me kind of go back in the slide to this kind of picture of what it looks like. And so there's there's a little bit of, extra stuff going on here. And it doesn't always directly track what's happening in EEG. There's also a little bit of a time delay. We tried to account for that between the electrical activity and this blood flow fMRI related measure. But yeah, we actually were not sure if we could do this on a more at the time resolved, at the scale of individual time frames. And I think it's promising that it's sort of, correlates with the EEG, but I think we can still improve it. Oh yeah,
Seems there are one more question.
yeah, okay, great. Yeah, I just see this now. So, the intracranial electrophysiological signals. Oh, yeah. So that's also a great question. We were assuming they were. The physiologists I collaborated with um, um, said that seemed to be a fine measure as well, and the local field potentials that were recorded intracranially. Um, in that study, we actually did use a slightly different spectral measure of alertness, only because the monkeys did have their eyes either open or closed. We couldn't control for that. And one effect of opening and closing your eyes is it modulates your alpha rhythm. And so when you are relaxed and you close your eyes, you can see this increase in a 10 hertz oscillation in the back of your brain. And so we didn't want our measure to kind of be confounded with those types of changes. And so we actually used a slightly higher beta band frequency in as part of this alertness index. And across the literature, it's actually very interesting is there's variability in what different studies use in terms of an electrophysiological measure of arousal. And this was very confusing to me at first. I was trying to sort through and figure out what we should use as a gold standard for training these models. But it does seem that most studies use some ratio of an alpha or a beta power band. So beta is up to like 1625 hertz or so. And they take a ratio with theta or delta bands, frequencies that are lower than 8 hertz. So thanks for the question.
And which measure was better, the fMRI or EEG for predicting behavior?
Oh, I think the EEG still did a little better than fMRI. That's a great question, actually. I should put that on the slides. Yeah. Oh, oh, great question, Julia. Yeah, did we do GSR, global signal regression, um, and calculated alertness based on this time series? So we tried this a little bit because I was curious as well, because these patterns that reflect alertness are actually quite global. And I believe that a lot of the predictive information is in the global signal. Now, the global signal itself, that's the average time course of all voxels in the brain. It's tricky because it doesn't necessarily reflect the same effects in every scan because it's just the average. But when there are a lot of changes in vigilance, that this is actually one contributor to the global signal. And so when we take that out, actually, I think it is removing some of the predictive information. Yeah, that's a great question. Okay. Yeah, so I just wanted to show a couple examples of how we might be linking these vigilance-related patterns with clinical measures. And so in this study, we looked at Alzheimer's disease data set. And one reason is that in Alzheimer's, early on, is known that key regions in the arousal pathway, the subcortical areas, such as in the brainstem and then the nucleus basalis of Meinert in the basal forebrain, these are thought to be some of the earliest to show degeneration. And so we were interested in, kind of exploring whether these patterns might be able to give us early markers of cognitive decline. And so we looked in patients with mild cognitive impairment who also had executive function and memory scores. And we tested some statistical models relating vigilance patterns in individual subjects to these measures. And here, so we did a little bit of a bootstrapping, or, kind of a we use a data-driven approach to get these vigilance patterns in individual subjects because they did not have concurrent EEG in this data set. And so what we can do is, even though we're using a canonical pattern to try to estimate someone's vigilance signal, we can try to back project this onto a subject's individual data to get a better picture of what that might look like in that person. And so that is the approach we used in this study. I also have students who are very interested in neuropsychiatric disorders and have been looking into the links with depression and anxiety. And so in this study, we used data from the Nathan Klein Rockland community sample. So it's not a patient sample per se, but measures of anxiety have been reported for every person. And so we can try to estimate a level of drowsiness from the fMRI data itself, and found that it does seem to correlate with the cross-subject scores on their state and trait anxiety index. And one thing we also did was again try to project the estimated arousal patterns, onto the in time courses, back onto the data of the individual subjects to see if there were any spatial variations that correlated with anxiety as well, and are finding some intriguing differences in regions like the hippocampus. So we're trying to follow this up and better understand it. Another application I'll show is in epilepsy, and this work is done with in collaboration with Dario Inglot and Vicki Morgan at Vanderbilt Medical Center. And one motivation for looking at epilepsy is that it's been found that patients with focal epilepsy may, they tend to experience issues maintaining their alertness during the day and other kinds of sleep-wake problems. And research from Dr. Inglot and others have found that this might be potentially arising from seizure-induced damage to circuits in the brainstem that are responsible for, these, widespread kind of, circuits that are regulating our vigilance. And so we were looking at connections between the subcortical brain areas and the rest of the brain. So what we do is we're deriving a measure of what's called functional connectivity. So we'll take a region and say a subcortical region in the brainstem, and we'll say, to what extent does that region signal correlate with other, the signals of other regions across the brain. Finding a difference in epilepsy patients between epilepsy and healthy controls. Interestingly, one thing we noted was that the patients seemed to be more drowsy overall during the scans. And this was reflected both in the behavioral measures that they did before the scans, as well as in some of their EEG measures. And so when we compare the patients and controls, we try to match them based on their brain state in order to make the cleanest comparison. Because if one group, is systematically more drowsy, and if we know their brain activity is different during the different state, then that might kind of, change the interpretation of what we're measuring. Okay, so in this next part of the talk, I'll talk about physiology, and really one thing that's been highlighted for me in exploring fMRI signals, and particularly in these different states of wakefulness, is how closely connected the brain and the body are. And so I like this illustration from work from Michael Chi's lab, which had focused on microsleeps. So you're watching a lecture, and every so often you might go to sleep briefly for a couple of seconds. This is a microsleep. And they recorded not only fMRI, but also EEG, heart rate, respiration. And they find that at the onset of this microsleep, there are changes not only in the brain activity, brain measure, fMRI, but also in the heart rate, in your respiration, and same when the person wakes up. So, these states are very, it's a whole system kind of physiology involved, not just the brain. We've been also interested in the changes in physiology that happen with slow drifts in wakefulness. So if you start out and you're becoming more drowsy, what we see in some people is that the variability in their heart rate tends to increase. There might be some changes in their respiratory patterns as well. And so, yeah, we're interested in the connections between this and the fMRI signal. And so in this work, led by Ben Gold, he actually found that as people are getting drowsy and falling asleep, the low frequency fluctuations in breathing and heart rate show a stronger contribution to fMRI data, including into the global signal, the whole brain average signal. So in other words, fMRI signals across the brain seem to they seem to have a closer connection to physiology when people are drowsy. So they tend to explain a larger fraction of the signal variations. And more broadly, there's growing amount of evidence that if we study the brain and the body together, that this can greatly enhance our understanding of individuals and disease. So in this example, this was a collaboration with Mara Mather's lab at USC, and they did this really interesting heart rate biofeedback studies. So people would, for several weeks, certain times of the day, they would be trying to consciously increase their heart rate variability, and they would get feedback from a program. And so, if you scan them before and after they do this kind of an exercise, then there's a change in the fMRI connectivity of brain networks that are related to functions like emotion and interoception. So we've been really interested. I would love to talk even more about physiology because it's something that I've become more and more interested in. But I'll show you a couple of snapshots of areas we've been working on here. One general challenge that we have in studying, say, cardiac and respiratory activity in conjunction with fMRI, is that even though it's useful in practice, we don't always get great signals. So this can be for any number of reasons. Like you might put a monitor on a person, say a photoplethysmograph, and they, if the person is moving their hand, then it kind of falls off, and you have to check it, put it back on. Sometimes people put on the devices, but don't necessarily check it till later and find out maybe there's something, there's some artifacts in the physiology data, and so. One great thing is that people are recording this data more and more. So the Human Connectome Project and a couple other of the large-scale initiatives to collect and share neuroimaging data have also included dynamic measures of a person's breathing and heart rate over that scan. But if we go to use it, then we need to spend time to try to quality check these measures, maybe try to correct artifacts whenever it's possible. And so one kind of engineering challenge we set ourselves was, can we actually infer some of the physiology directly from fMRI in a way that's kind of aligned with how we were trying to estimate people's vigilance level. And we are definitely not the first to do this. There's actually been quite a bit of work in this area trying to infer different features of physiology from fMRI. So some looking at maybe the beat-to-beat cardiac waveform or the phase of the respiratory signal and so forth. And here what we focused on was a more low frequency amplitude of somebody's breathing or their heart rate over time. And so what we did was in our first study, we looked at data from the Human Connectome Project, which, as I mentioned, does have physiological recordings. And the problem setup is essentially you have your fMRI data, you can extract the activity signals, time courses from different brain areas, and we want to say, can we actually then produce a time course of how deeply the person is breathing over time? So like, this looks a little bit like the ins and outs of someone's like every breath, but actually I should emphasize that this is more of like an envelope of somebody's breathing. So we naturally kind of fluctuate between taking more deep breaths or more shallow breaths and so forth. And so we started with a few simple approaches, convolutional neural network, linear models, and got a range of performance. Some of the good ones I show here. So this is a close correlation between the estimated and the ground truth or measured physiological recording. We screened the physiological recordings to develop our initial set to, just to make sure we were not including really corrupt ones to use as the ground truth for training. So, and then, yeah, but there's a range, and our median was about 0.5 or so, but kind of was, interesting to see how that, there is a lot of shared information between brain fMRI and the respiratory signal, in that we can kind of track these changes based on the fMRI signal. And then we wanted to do a couple more things to extend this. So A, we wanted to see if we can increase the performance, and B, we wanted to see if we could simultaneously reconstruct breathing and heart rate time courses from fMRI, which would give us nice kind of more end-to-end solution to do both signals if we needed those. And so Rosa Byrek has really taken this to new heights, using multiple brain atlases as regions of interest, developing and testing other deep learning architectures for doing this type of task. And these results are from a bidirectional LSTM model. And so here's an example of the performance which we're able to increase. So this is one of the good scans, and showing the measured and predicted respiratory variation as well as heart rate, along with the distribution across the group. So we got our median results to around Pearson correlation between the measured and ground truth signals at closer to 0.6, 0.7 here. And then, we wanted to see if we were going to distribute this as a tool for inferring physiological waveforms from fMRI. We wanted to make sure that it could generalize across different settings, different acquisition parameters, for acquiring fMRI data, different task conditions, and so forth. And so we also tested the generalization to different tasks people are doing in the human connectome data set. So there's some scans where people are doing an emotional task, working memory task, and so forth. If we don't retrain and we directly just take the model trained on resting state data, which is the main results I showed before. Resting state, if you're not familiar yet, they're just closing their eyes, not doing anything else in particular. Actually, sorry, in this case, they're open and just looking at a fixation cross. Then it is doing better on the type of condition it's trained on in a cross validation, but also, generalization is not too much worse. It also seems to work on an external data set that was acquired with quite different acquisition parameters. And so we're now, in current work trying to see if we can make, the performance better, as well as seeing if it generalizes to other populations. And we recently did an investigation in aging. We're also interested in other reasons, or for other reasons, in studying connections between physiology and fMRI and aging as well, because it seems like there could be some changes. And so this is kind of illustrating that we're able to also, to some extent, predict these respiratory signals in older adults as well. And Shiyu and Zian have done an amazing job extending the models to be able to better handle variation across age. So we're trying to collect these tools into a a platform, and hopefully like have them be usable to other groups and studies. Rosa has been leading this effort, so we have been putting our tools for this deep learning inference of physiology on this site, and we are also developing other tools that we found to be quite useful in these studies, in terms of even just if physiology is recorded, quickly being able to quality check these, being able to maybe fix certain artifacts in the physiological waveforms, and also being able to do, both in terms of a manual and interactive platform, as well as a more automated tool where, say, you were input, your hundreds of physiological recordings, and can it do an initial good sorting of whether or not the signal is actually usable or not. So we are trying to, yeah, hopefully be able to make tools that are useful for the community. So, very last study I'll show is that, I've been actually, I separated these into two different sections, not talking so much about the interconnection between them, but uh, as we did briefly mention, the states of vigilance and the physiology of your body is very highly connected. And this study was led by a collaborator, Taylor Bolt, who brought together many different data sets that each have fMRI and at least one measure of person's arousal or physiology. And overall, is able to find that there's a major component of the spatiotemporal variance in fMRI that appears to correlate with these multiple physiological and vigilance signals. And so I think that these different signals don't always go together, but there is a lot of common modulation as well. And I think this really further underscores the unification of the brain and body. So I'll just summarize here. fMRI signals are very rich in terms of their spatial patterns, also their dynamics. It seems that we are able to extract information about internal states of vigilance and autonomic physiology from fMRI signal dynamics, and that this could be useful, for better understanding our data, doing more precise imaging and interpretation, as well as new avenues for biomarkers. And yeah, I would like to thank all the people in our lab and collaborators who've been involved in this work. And thanks for your time.
Thank you. Thank you very much for a great talk. We'll wait for questions. In the meantime, I had one. You mentioned aging, and I was trying to keep track of aging throughout. So as you know very well, the story is that variability goes down in aging, and that is correlated with behavioral measures. But it seemed you were saying that the variability of the neural signal correlated better with the physiology in older people.
Oh, yeah.
And so I was trying to track all these things together. So what happens to physiological variability in aging? What happens to fMRI variability? The correlation is higher. How is that all tying together? And can we explain aging variability by something else other than neural variability?
Oh, great question. So I should first clarify that we didn't show so the physiological variance that increased was during drowsiness, not necessarily during aging. So
Ah,
yeah, so I presented those slides fairly close together. So that was my, I was not sorry for
but
not being more
but
clear on
the
that. So.
explanatory ability of the physiology increased in aging, right? I think that's what you were saying, if I understood.
Oh, so I actually didn't show data on that. We are kind of exploring that at this moment. We found that so there are two relevant studies in here. So one was the physiology variance explain increase in drowsiness, but the other is that in the group where we're looking at Alzheimer's, early stages of Alzheimer's, then the spatial patterns, there were regional differences in the spatial patterns
Okay.
that
Okay. No, I
correlated.
apologize.
Oh yeah, no worries at
I
all.
filled in that connection in my head. I
Yeah.
was starting with our older people sleeping more in the scanner. This is why I was then trying to connect the drowsiness level of older
Yeah.
versus younger people and trying to explain the variability through the drowsiness connection. No, no,
But
now I'm
actually,
back on track.
yeah, no, this is a great question because we're also doing some studies on this at the moment. So we it actually does seem to relate to their brain state, like whether whether or not we see physiological variance increase or decrease in older adults, and so we have depending on the data we look at and what their state is, we can see either reduced variance in the physiology in older adults, but also, yeah, it does seem to be connected to whether or not they're sleeping or not. So didn't show any slides yet because we're still trying
Yeah.
to check our analyses and make sure everything is correct. But yeah, we do see some trends toward decreased variance explained in older adults, but some differences in the dynamics. So
Okay.
yeah,
Okay. So, but these are linked, right? Because it's always been the
yes,
issue, right?
yes.
This aging variability, what is the source of? Because it seems replicable, but it also seems like what is the actual source, right?
Exactly. Yeah, and I think that's important source to account for and yeah, making this interpretation because definitely even if the two are, like yeah, yeah, you definitely could see difference, more drowsiness in some of the older adults, and
Yeah.
that will indeed correlate with stronger physio
Yeah.
fMRI
Yeah.
correlations. So yeah.
Yeah. Other questions in the chat? Raise your hand, interrupt. Yes, Ryan.
Hi Katie. I really enjoy your talk. It was really interesting. So I have two questions related to the first part about vigilance state. So I saw that when you look at how the spatial pattern linked back to the alertness index, you show that there's some like cortical like the cortical networks correlations are kind of tracking correspond to the different clusters of drowsiness. So so because you only show like the cortical networks, I'm wondering, did you consider the brainstem regions of interest in like and using those too? Yeah.
Yeah, yeah. Thank you so much for asking that. And so yeah, in that study, we did mostly focus on the cortical areas. There are probably brainstem areas in those maps that I haven't actually like yeah saw characterized in that particular study. But we are doing other studies on the connectivity of brainstem regions across um, these levels of alertness, and we can see some differences. So it seems that the many nuclei are um, more correlated with the cortical patterns that I showed during drowsiness,
Um,
say like more of the sensory cortices, whereas during more alert phases, they seem to be more correlated with networks like the default mode network and salience network. And so there definitely seems to be some, yeah, they're part of these systems that change and possibly responsible for them. So I think the brainstem is really interesting to look at here.
I see. Okay, perfect. And then another question is that I know you're really interested in the fast temporal dynamics and how like, alertness kind of varying like in those temporal scale. But I'm just curious, does like the individual's like functional connectivity like predicts like the kind of the like during the whole scan, like on average, I suppose some individuals will spend more time becoming sleepy in the scanner versus some
Yeah.
people who's better at keeping their alertness during the scan. So I'm wondering if there's any brain activity that predicts like those on average who spends more, like who are more sleepy and who are more vigilant.
Yeah, so one thing we've noticed is that the and others too, like Thomas Liu and UCSD, like they've also shown that the kind of variation in the data is one predictor. So the time courses, fMRI time courses seem to increase their fluctuation amplitude as people are becoming drowsy. And if in the study I showed where we connected estimated drowsiness with a measure of anxiety, we estimated their drowsiness by looking at the standard deviation of our alertness time course, actually, because I think that as they're becoming drowsy, they might have more modulation of this kind of alert drowsy, dynamics. And so that ended up being one indicator. There also tends to be more global correlation when people are drowsy, and I think possibly for both neural and physiological vascular reasons. Partly linked with the fact that the variance in those respiratory and cardiac signals are also, you know, correlating with that, as is neural wide-scale line neural changes linked with arousal fluctuation. So yeah, those are two kind of the signatures that I could point out. But I think that connectivity is also one. We'll show that as well.
I see. Thank you so much.
Leon?
So I missed the beginning of the first few minutes, and maybe you mentioned this, and if so, excuse me, but it's very interesting. I don't know very much about this. When you take an fMRI of somebody, are they sticking their head in a machine, and is that machine making lots of noise?
Yes, yes, it is.
So can you actually get drowsy under that environment?
Believe it or not, yes. The noise, although it's loud, it's very repetitive, and we give them earplugs and a blanket, and I actually have more trouble keeping them awake, believe it or not. I thought I would have the opposite problem, but in our data sets, it's almost harder, especially in these rest scans where we just have them close their eyes and relax. We're not giving them anything to do, which would more likely keep them awake. But yeah, in these scans where we have them just close their eyes and maybe just do some low level like auditory stimulus, they can fall asleep in a couple minutes. And there's also been a study by another lab that tried to like track how long it took people to fall asleep in the scanner, and it turns out that within like 5 or 10 minutes, so many people were sleeping. So it's unusual, but yeah, I definitely get sleepy.
And when you talk about a time point, I thought that when taking a scan, it takes, 20 minutes or something, but you
Oh
were
yes,
talking about fluctuations. I wasn't sure what the time scale of your fluctuation is,
yeah.
but I thought it was rather short. So how do you get, how long is it when you
Oh
talk
yes,
about time point? How long is it?
yeah. So, what I meant by a time point here, and I know that it's used in different contexts depending. So here, the scan is like 20 minutes or so, but a time point I here referred to the length of time it would take to acquire one 3D volume worth of data, which is 2 seconds, around 2 seconds. In this most of this studies, 2.1 seconds, I believe.
I see. And that final question, I know a lot about heart variability and respiration. And when you're doing a comparison with that, what are you actually comparing it to when you're talking about heart rate variability?
So we calculated a time course of the heart rate variability, and we would compare it to the
Oh,
time.
what do you mean by heart rate? What
Oh,
is heart rate variability?
oh, what heart rate variability? Okay,
You're
yes.
actually using
So
testing heart rate variability?
right. So when we made our time courses here, we actually just looked at a simple like beat to beats kind of difference. It was a kind of short um, window of time where we captured a kind of mean beat to beat differences. So that's more just like in the tracking the heart rate, I would say, in a moving windowed fashion. Um, when we in other studies, not ones I've shown here, but we've used like tried to use other measures like the root mean squared successive RMSSD and standard deviation and
It's
other
still not
measures.
clear exactly what it is that you're
Yeah.
that
So
that
mhm.
that you're taking.
Yeah, so here we just take a short, like 6 second window around centered on every fMRI acquisition. We look in the physiological data and we take the um, beat to beat interval length, um, find an average, and then invert it to convert it to a heart rate. Um, yeah, so that was the dynamic heart rate time
Okay.
course we
Okay.
calculated.
Cool. Any other questions? If not, then once again, thank you so much for this very inspiring and interesting talk. And yeah, and Katie, I'll write to you as in a separate email about the waves. Great. Thank you so much. Thanks everybody for coming as well. Bye.
Thank you.